Hello, this is a video lesson on ribosomes and protein synthesis. And the objective of this video is to have you as the student be able to construct your own polypeptide from an mRNA uh, strand and or list the steps of translation and the mRNA's journey through a ribosome in the creation of a polypeptide. First, we're going to start with the prerequisites for translation. The first step in decoding the genetic message is to transcribe a nucleotide base sequence, or transcription, into mRNA. And that was in a previous lesson on transcription that we saw how that works and how you get a strand of mRNA. Also, before we start talking about this, you should also know what a polypeptide is, which is a long chain of amino acids called a polypeptide, obviously, um, or a protein. The sequence of these amino acids determines its shape and therefore also its function. And that's speaking to how a protein folds as it's coming out of the ribosome. Will determine its shape. Its shape determines how it catalyzes reactions or other various functions within the cell. The genetic code. RNA contains 4 different bases, as previously discussed: adenine, guanine, cytosine, and uracil. These make up the basic alphabet of the genetic code. If you were thinking of the genetic code as a language, these would be the letters. The codon would be the words of a genetic code. Basically, mRNA is read in order, 3 consecutive bases at a time. And a specific codon only codes for one amino acid. So you have 3 bases and a codon, and those code for one amino acid. And I will get into that a little bit more here in a bit. How to read a codon. There are 4 bases in RNA. As we already discussed, this allows for 64 possible codons because codons consist of 3 bases. And as you see here, I have a simple math problem that shows you why it is 64 possible combinations of codons. You have 4 base pairs times 4 base pairs times 4 base pairs, which is the triplet codon, which equals 64 possible combinations. So that means there are many more combinations than there are actual amino acids. There are only 20 amino acids. So what this means is that most amino acids actually have multiple codons which represent them. So let's see, in the case of serine, it has UCU, UCC, UCA, and UCG codons code for it. So it has 4 possible codons that will all code for serine. However, only one codon can represent one amino acid. So a codon can only represent one amino acid. It can't represent multiple amino acids. It can't be CGA representing arginine and leucine. It just does not work that way. Here we have a coding chart, and this is how you will be able to translate or understand translation of an mRNA code. As you see here, the inside circle represents the first base in the codon, the middle base is the second base, and the outer base is the last base in a triplet codon. Um, and what you see is that each of these um, represent another amino acid. You have the whole list right here, so it makes it quite simple to be able to translate an mRNA strand into what it will be as a polypeptide. Also important in codons are the start and stop codons, which tell the ribosome where to start and end translation. The start codon is AUG. AUG codes for methionine, so methionine will always be the first uh, amino acid in a polypeptide chain. Sometimes it's removed later. Actually, the majority of the time it's removed later. But the first amino acid in a polypeptide chain will always be methionine. There are also three separate stop codons: UGA, UAA, and UAG. These do not represent any amino acids. They actually terminate translation by putting a nonsense protein in place of a tRNA at the first site, which I will get into more later. It'll make more sense here in a bit. Finally, we are on to translation, the actual process by which mRNA turns into or codes for polypeptides or proteins. Translation is the process by which ribosomes use the sequence of codons in mRNA to assemble amino acids in a polypeptide chain. And there are three phases in this process: initiation, elongation, and termination, which I will talk about in the upcoming slides. Also, before we start, you should know that tRNA brings an amino acid to the ribosome as specified by mRNA code. Now, how this works is the codon in mRNA is complemented by an anticodon in the tRNA. So basically there's a complementary triplet code on the tRNA which is carrying the amino acid. So they pair up by base pairing in the ribosome, and that is how the mRNA will code for a specific amino acid. First we have initiation. The ribosome attaches to an mRNA molecule and moves along the molecule until it reaches an AUG, as you can see in that left side picture. tRNA with an anticodon complementary to AUG, which contains methionine, will then move to the middle site of a ribosome, also known as the P site. It will then complementary base pair to that, and we are ready to move on to the next step. Second step is elongation. This is comparative to an assembly line. Amino acids are added to a growing polypeptide chain as the ribosome moves codon to codon through an mRNA strand. A new tRNA complementary to the second codon will move into the first site of a ribosome, also known as the A site. Now, a peptide bond is formed between the first and second amino acids. Now, as you can see, the picture at the bottom, if you go from the top, that's where I started, you have methionine in the P site. Go down. A second amino acid or tRNA comes into the A site. The third step is that a peptide bond is formed between methionine, which was the AUG, the first start codon, and the new tRNA or amino acid brought in. Simultaneously, the UAG, the tRNA, which codes for UAG, the anticodon to that, will release its methionine, so there's only one bond between methionine and the second amino acid. And as you can see in the third step, or in the fourth step, the first tRNA is ejected from the cell, or ejected from the ribosome, and you're ready for a new tRNA to move into the A site and for this process to continue. Finally, we have termination. And this is when the peptide chain continues to grow until it reaches a stop codon on the mRNA molecule. When this occurs, both mRNA strand and the polypeptide are released, thus completing the process of translation. Now, as you can see in the bottom picture down below, the left picture starts with the last tRNA in the polypeptide coming in, and you have the stop codon next. Now the release factor will move into the A site. It will basically simulate a peptide bond between that last amino acid and itself, so the tRNA will release. Now this happens, and the polypeptide is basically left out into the cytosol or cytoplasm, the innards of a cell. And at this point, everything is released. The release factor, the protein or polypeptide chain is already free floating, and the mRNA is also released. As a review, we're going to look at the roles of RNA in translation. First, we have mRNA, which carries the coded gene, and that's a product of transcription, as discussed in a previous video lesson. tRNA, which delivers the right amino acid based on the mRNA code, and we also discussed that. It's when there's an anticodon, which is read by the tRNA, or by the ribosome, and it is matched up with its mRNA complement, so that you have the right amino acid coming in. And thirdly, ribosomes are composed of 4 different rRNA molecules and roughly 80 proteins. They translate the mRNA code. So you have 4 different ribosomal RNAs and 80 proteins that come together to form the small and large subunits of the ribosome. And that is it.